Human genome research has a rich history that dates back to the mid-20th century. One of the key milestones in this field was the discovery of the structure of DNA by James Watson and Francis Crick in 1953. This breakthrough laid the foundation for understanding the genetic code that makes up the human genome. In the following decades, advancements in technology, such as the development of DNA sequencing techniques, revolutionized the study of genetics. The Human Genome Project, HGP, launched in 1990, marked a significant turning point in human genome research. This international collaborative effort aimed to map and sequence the entire human genome, consisting of approximately 3 billion base pairs. The project was completed in 2003, providing a comprehensive reference for the genetic makeup of humans. It not only enhanced our understanding of genetic diversity, but also paved the way for further research in genetics and personalized medicine. Since the completion of the HGP, research in human genomics has continued to advance rapidly. The development of high-throughput sequencing technologies has enabled scientists to sequence genomes more efficiently and cost-effectively. This has led to the discovery of numerous genetic variations associated with diseases and traits, providing valuable insights into the genetic basis of various conditions. Moreover, the field of epigenetics has emerged as a key area of study in human genome research. Epigenetic modifications such as DNA methylation and histone acetylation play a crucial role in regulating gene expression and can influence various biological processes. Understanding these epigenetic mechanisms has opened up new avenues for exploring the complexity of the human genome. Overall, the history of human genome research is a testament to the remarkable progress made in understanding the genetic blueprint of humanity. From the pioneering work of Watson and Crick to the completion of the Human Genome Project and beyond, each milestone has contributed to our knowledge of genetics and holds promise for the future of genetic engineering and personalized medicine. Genetic engineering techniques encompass a range of sophisticated tools and methods that allow scientists to manipulate and modify the genetic material of organisms. One of the fundamental techniques in genetic engineering is recombinant DNA technology, which involves the insertion of foreign DNA into an organism's genome. This process enables researchers to introduce new genes or modify existing ones, leading to the production of genetically modified organisms, GMOs, with desired traits. Another key technique in genetic engineering is gene editing, which allows for precise modifications to be made to the DNA sequence of an organism. One of the most widely used gene editing tools is CRISPR-Cas9, a revolutionary technology that enables targeted changes to be made to the genome with unprecedented accuracy and efficiency. CRISPR-Cas9 works by guiding a molecular scissor-like enzyme, Cas9, to a specific location in the DNA sequence, where it can then cut the DNA and introduce desired changes. This technology has revolutionized the field of genetic engineering and holds immense potential for applications in agriculture, medicine, and biotechnology. In addition to recombinant DNA technology and gene editing, other genetic engineering techniques include gene cloning, polymerase chain reaction, PCR, and gene synthesis. Gene cloning involves the creation of multiple copies of a specific gene, which can then be studied or used for various applications. PCR is a technique that allows for the amplification of specific DNA sequences, enabling researchers to generate large quantities of DNA for analysis. Gene synthesis, on the other hand, involves the artificial assembly of DNA sequences, allowing for the creation of custom-designed genes. These techniques have revolutionized the field of genetics and have paved the way for groundbreaking discoveries and advancements in genetic engineering. With the continued development of new technologies and tools, the possibilities for genetic engineering are endless, offering exciting prospects for the future of science and biotechnology. Ethical considerations play a crucial role in the field of genetic engineering, as the ability to manipulate the genetic material of organisms raises complex moral and societal issues. One of the primary ethical concerns surrounding genetic engineering is the potential for unintended consequences. Modifying the genetic code of an organism can have far-reaching effects, both positive and negative, on the environment and other species. For example, the release of genetically modified organisms into the wild could lead to ecological disruptions or the spread of engineered genes to non-target species. Additionally, there are concerns about the long-term impact of genetic modifications on biodiversity and ecosystem stability. Another ethical consideration in genetic engineering is the issue of informed consent. In the context of human genetic engineering, questions arise about the autonomy and rights of individuals to make decisions about their own genetic information. The use of genetic testing for predictive purposes, such as identifying predispositions to certain diseases, raises concerns about privacy, discrimination, and the potential misuse of genetic data. Furthermore, the concept of genetic enhancement raises ethical dilemmas regarding the boundaries of what is considered acceptable in altering human traits and characteristics. The ethical implications of genetic engineering extend beyond scientific and technical considerations to encompass broader societal values and beliefs. As we continue to advance in the field of genetic engineering, it is essential to engage in thoughtful dialogue and ethical reflection to ensure that these powerful technologies are used responsibly and in the best interests of society as a whole. Recent years have witnessed remarkable discoveries in the field of human genomics, shedding light on the intricate complexities of our genetic makeup. One of the groundbreaking findings is the identification of genetic variants associated with complex diseases, such as cancer, diabetes, and cardiovascular disorders. Through large-scale genome-wide association studies, GWAS, researchers have uncovered thousands of genetic loci that contribute to disease susceptibility, providing valuable insights into the underlying mechanisms of these conditions. For instance, studies have revealed specific genetic mutations that increase the risk of developing breast cancer or influence an individual's response to certain medications. These discoveries have paved the way for personalized medicine approaches that take into account an individual's genetic profile to tailor treatment strategies for better outcomes. Another significant advancement in human genomics is the exploration of non-coding regions of the genome, once considered junk DNA. It is now evident that these non-coding regions play crucial regulatory roles in gene expression and cellular functions. Researchers have identified functional elements such as enhancers and long non-coding RNAs that control gene activity and contribute to various biological processes. Understanding the role of non-coding DNA has opened up new avenues for studying gene regulation and disease mechanisms. Moreover, recent studies have highlighted the importance of genetic diversity and population genetics in shaping human evolution and adaptation. By analyzing the genomes of diverse populations worldwide, scientists have uncovered genetic variations that reflect historical migrations, environmental adaptations, and selective pressures. These findings not only enhance our understanding of human diversity, but also have implications for personalized healthcare and disease prevention. In addition to disease-related research, recent discoveries in human genomics have expanded our knowledge of rare genetic disorders and syndromes. Advances in sequencing technologies have enabled the identification of novel disease-causing mutations in rare genetic conditions, providing hope for improved diagnostics and targeted therapies. For example, the use of whole exome sequencing has facilitated the diagnosis of rare genetic disorders in patients with undiagnosed conditions, leading to better management and treatment options. Furthermore, the field of epigenetics has emerged as a key area of research in human genomics, offering insights into how environmental factors and lifestyle choices can influence gene expression and health outcomes. Epigenetic modifications, such as DNA methylation and histone acetylation, have been linked to various diseases including cancer, neurodegenerative disorders, and metabolic conditions. Understanding the interplay between genetics and epigenetics holds promise for developing novel therapeutic interventions and preventive strategies. Overall, the current discoveries in human genomics underscore the dynamic nature of genetic research and its profound impact on healthcare, personalized medicine, and our understanding of human biology. Genetic engineering has revolutionized the field of medicine, offering innovative solutions for diagnosing, treating, and preventing a wide range of diseases. One of the most significant applications of genetic engineering in medicine is the development of gene therapy, a cutting-edge approach that aims to correct genetic defects and restore normal gene function. Gene therapy holds promise for treating genetic disorders, such as cystic fibrosis, muscular dystrophy, and sickle cell anemia, by delivering functional genes to replace or repair faulty ones. In recent years, several successful clinical trials have demonstrated the potential of gene therapy in providing long-term benefits to patients with genetic diseases. Another key application of genetic engineering in medicine is pharmacogenomics, which involves using genetic information to personalize drug treatments based on an individual's genetic profile. By analyzing genetic variations that affect drug metabolism and response, healthcare providers can optimize medication regimens to improve efficacy and reduce adverse effects. Pharmacogenomics has the potential to revolutionize drug development and prescribing practices, leading to more tailored and effective therapies. Furthermore, genetic engineering plays a crucial role in the field of cancer treatment through precision medicine approaches. By analyzing the genetic mutations driving cancer growth, researchers can develop targeted therapies that specifically inhibit the molecular pathways involved in tumor progression. This personalized approach to cancer treatment has led to significant advancements in improving patient outcomes and survival rates. Additionally, genetic engineering has enabled the production of recombinant proteins and monoclonal antibodies for therapeutic purposes. Biopharmaceuticals, such as insulin, growth factors, and antibodies, are produced using genetically engineered organisms, offering novel treatment options for various diseases. These biologics have revolutionized the treatment of conditions like diabetes, autoimmune disorders, and cancer, providing patients with more effective and targeted therapies. Overall, the applications of genetic engineering in medicine continue to expand and evolve, offering new possibilities for personalized healthcare, disease management, and drug development. The future prospects of genetic engineering hold immense promise for revolutionizing healthcare, agriculture, and biotechnology. One of the most exciting areas of advancement is the development of gene editing technologies such as CRISPR-Cas9 that enable precise modifications to be made to the genome with unprecedented accuracy and efficiency. These tools have the potential to revolutionize the treatment of genetic disorders by correcting disease-causing mutations at the molecular level. In the coming years, we can expect to see gene editing therapies becoming more widely available, offering new hope for patients with previously untreatable genetic conditions. Another exciting prospect in genetic engineering is the field of synthetic biology, which involves designing and constructing biological systems for specific applications. Researchers are exploring the creation of synthetic organisms with novel functions, such as producing biofuels, drugs, and materials. Synthetic biology has the potential to transform industries and address pressing global challenges, such as environmental sustainability and healthcare. Furthermore, advances in personalized medicine are set to reshape the healthcare landscape, with genetic information playing a central role in tailoring treatments to individual patients. By analyzing an individual's genetic profile, healthcare providers can predict disease risks, optimize drug therapies, and recommend lifestyle interventions for better health outcomes. The integration of genetic data into clinical practice is expected to lead to more precise and effective healthcare delivery, ultimately improving patient outcomes and reducing healthcare costs. In agriculture, genetic engineering offers opportunities to enhance crop productivity, nutritional value, and resilience to environmental stressors. Scientists are developing genetically modified crops that require fewer pesticides, have increased yields, and are more nutritious. These innovations have the potential to address food security challenges and promote sustainable agriculture practices. As we look to the future, the possibilities for genetic engineering are vast and transformative. From personalized medicine and gene editing therapies to synthetic biology and agricultural advancements, genetic engineering is poised to drive innovation and impact diverse sectors of society. By harnessing the power of genetic information and technology, we can unlock new solutions to complex problems and improve the quality of life for individuals around the world. Genetic engineering faces challenges and limitations, including off-target effects in gene editing that can lead to unintended genetic changes. Ethical concerns surrounding the use of genetically modified organisms and the potential for genetic discrimination also pose significant hurdles. Additionally, the complexity of the human genome and the interactions between genes make it challenging to predict the outcomes of genetic modifications accurately. Technical limitations, such as the delivery of gene editing tools to target cells and the efficiency of gene modification, remain areas of active research and development. Overcoming these challenges will require continued innovation, rigorous safety assessments, and thoughtful consideration of the societal implications of genetic engineering. In conclusion, genetic engineering holds immense potential for transforming healthcare, agriculture, and biotechnology. From gene editing therapies to personalized medicine and synthetic biology, the future of genetic engineering is filled with exciting possibilities. However, challenges such as off-target effects, ethical concerns, and technical limitations must be addressed to ensure the responsible and effective use of these technologies. As we continue to advance in the field of genetic engineering, it is essential to prioritize safety, ethics, and societal considerations in our research and applications. Now I would like to open the floor for any questions you may have regarding genetic engineering, its applications, challenges, or future prospects. Your questions are valuable in fostering a deeper understanding of this rapidly evolving field.